Ну, сегодня мы на самом деле собрались, чтобы еще раз проговорить весьма сложную, как с технологической точки зрения, так и с этической точки зрения, проблему, связанную с генной терапией. Вот здесь вот висел до моего портрета замечательная совершенно тема «Комплекс Бога». И в действительности, манипулируя с геномом, насколько быстро мы приближаемся к тому, чтобы противоречить Создателю нашему, и насколько быстро мы можем изменять, собственно говоря, то, что является венцом творения человека. И для того, чтобы обсудить сегодня эти тематики, мне бы хотелось пригласить на сцену двух спикеров, которые помогут в рамках открытого интервью эту тему обсудить и, возможно, ответить на ваши вопросы, касаемые как этических проблем, так и технологических проблем. Я бы хотел пригласить на сцену господина Джуона Хуа-Пан. Это специалист по генной терапии и профессор Университета Уэйна. Мистер Пан, welcome to the stage. Вторым участником сегодняшней панели у нас является господин Шон Эйнсворт. Это основатель компании RetroSense Therapeutics и генеральный директор компании RetroSense Therapeutics. Это уже компания, которая инвестирует непосредственно в технологии, связанные с генной терапией. Шон, please welcome to the stage. Наше интервью. Я буду говорить по-английски, поэтому те, кому нужно, пользуйтесь синхронным переводом. Я бы хотел начать с вопроса к аудитории на самом деле. Я прошу поднять руки тех коллег, которые имеют непосредственное отношение к биологии, к генам, кто занимается этой проблематикой. Я бы также хотел спросить, кто здесь из присутствующих в зале занимается вопросами биологии и медицины, кроме тех, кто занимается непосредственно геномикой? Отлично. Ну что ж, у нас вполне с вами... So, we've got the auditorium, which is more specialized. You see a lot of people raise the hands. That means the people have some experience either with genomics or with medicine at least. So, we can be more specific, which is good. Okay? Yeah. Nevertheless, I would like to start with a very basic question. Because for the moment, the stereotypes about gene therapy is completely different in different hands. Some people believe that's genome editing, which allows us to change the features of future child. Some people believe that changing gene, we can fix some very tough diseases, not on the system diseases, not only on the organ level, but on the organ of the health complete human body. But just talking about the basic technology, when we are talking about gene therapy today, which can be implemented in the real medicine as well, which already came out from the scientific labs to the healthcare system, what do you understand, Professor Pan, as the gene therapy?
Okay, the gene therapy basically, the simplest way to say is to use a gene to treat the genetic disorder or disease. Okay, that's the simplest definition. Now, also, so the basic for the gene therapy, basically, we know that the information which is guiding the development of the human body, and also maintain the health of the body, and also determine the future and the characteristics of each individual, is stored in the DNA. Okay, that's. And so that's basically inside the nucleus, okay, for each individual cell. So actually, most of people already know this one, but I just give briefly in case someone don't know. So the DNA basically is a human contain about 4 million, okay, the nucleotide, the basic the unit to form the DNA. So basically, it's very simple. Only have 4 types. So it's a simple abbreviated as ATCG. Okay, that's the coding nucleotide for the DNA. Now, so basically now the DNA, a portion of the DNA, that's called coding DNA. So they're going to eventually translate to the protein. So you know the protein is the important building block for the body. Okay, so we already know right now about 20 to 30,000 the segment of this DNA, which is called a gene. Okay, this gene eventually to transfer to the protein. That's very important for the building block for the. So now, so in mutation, okay. So in this nucleotide, so I talk about CG. So because from the DNA to transfer the protein, basically, is the protein basically is the polymer of the amino acid. Okay, that's either long or short. Now each amino acid is encoded by 3 nucleotides. So from the DNA. Okay, that's how this relation between the DNA to the protein. Now, right now, also people know many diseases could trace back to cause due to the mutation in the DNA level. So the idea of the gene therapy, okay, to treat this inherited disease, okay, that's we could be fixed by directly fix the source, okay, in the DNA level. Okay, that's the basic of the gene therapy idea. So now also, why at this level is made it possible, we can use the gene therapy. Basically, this depends on a number of things. Now, I think the first concept of the gene therapy is started in the 1970s. Because at that time, it's become possible due to the molecular technology development. So they can do the recombinate, so you can swamp of the DNA, okay, to make changes. And also, they have sequencing technology developed, so people can sequence the genome. So basically, at the time of 2003, the whole human genome have been sequenced. Okay, that's we have about 4 million nucleotides. And also another one is scientists did more study, okay, to figure out each disease, okay, to trace back to which mutation in the DNA. Okay, that's also very important. So you can know either this could involve one gene or multiple genes. And the final one is also the delivery technology also become more developed. So basically, right now we can use the more efficient ways to use the virus. So we can package the DNA we want to correct to the virus, then transfer it into the cell. So all these put together. So right now, okay, we are really in the time, so we can move to technology in the very rapid pace. So I think in the last year, they talk about the gene therapy right now is have more than 2,000 gene therapy in the clinical trial. Okay, and I think in the U.S., I think there's no FDA approved. Okay, all still in the clinical trial stage. Yeah, but I can proceed, and in the future, okay, this in the future, in a few years, okay, this become really can be go to the medicine. Yeah.
Okay, thank you very much. We got the idea how it works, but what are the best application, or maybe not even the best application, but application we do expect in clinic first with this type of the approach? Which nosologies, I mean, which kind of diseases, and where is it would be more effective based on the advantages of gene therapy you mentioned?
Okay, I think currently the gene therapy would be in the best scenario will be the disease is caused by a single gene or a few genes. Okay, that would be more efficient. Okay, so if the disease is caused by a lot of genes, so I think they still have a technology the difficulties to do this one. And although I think this technology continue to develop, even in the last few years, so people probably know is called a CRISPR-Cas9 system. Okay, so in that system was developed, really can allow people, the scientists can to cut in the precise location and also in the multiple places you can repair the defect gene. And also I think because I'm working on in the eye, okay, we are particularly interested in for treating the blindness, okay, particularly. So I think the blindness and Sean also talk about. So we developed this optogenetic approach as one potential treatment for people lost vision due to the degeneration photoreceptors. So right now, I think this is very promising. The company RetroSense already moved to the clinical trial.
So maybe you would tell us a couple of words more about your development and about your project, because for me it looks amazing. In fact, what you are doing and what you do expect to have as a first patient next few days, as Sean mentioned behind the scene, you basically, with this technology, you're going to make blind person got the vision back. Is it correct?
That's what I hope. Yeah.
No, according to the experience, we have to. Come on.
That's the expectation.
That's the expectation. We
Yes.
really believe in this expectation. We really want to see this.
Most
But
definitely.
talking about the technology, maybe a couple more words about the technology you developed. And the next part of the question, after the technology is clean, how it would look for the patient. I mean, what would be the patient experience? The technology first. What do you do?
Okay, the technology. Okay, even before talk about my particular optogenetic gene therapy, I want to talk about more general. Okay, the gene therapy can be divided in the multiple categories. Okay, one is the replacement gene therapy. So basically, so if one gene is a defect, and so then this defect of the gene could be loss of the protein, so the protein loss of function. So in that case, we have to repress the normal gene, okay, to make the normal protein. That's one. Another kind of gene therapy is a certain protein, basically, when due to the mutation or something, they become gain of function. So basically, they are overexpressed. Okay, that's also cause a problem. So that's when, in that case, we have to delete this gene. Okay, that's one. And also now the technology belongs to what we're using right now. Basically, is not even this first two. Basically, we put a foreign gene into the cell, okay, to make the cell the function which they're not there before. So basically, I'm going to talk about more this one. And basically, we use the gene from the green algae. Actually, even is not remotely related to the human gene. Okay, so put it in the retina to try to restore vision.
I cannot be in silence. You know, listening this. Can we develop it further? Sorry for provocative one, but for example, if we go this way, can we make a night vision for the person available?
Yeah, that's possible because actually we also try to improve this particular gene to make the protein which you can sensitive to the infrared, okay, or in the other spectrum UV light.
Infrared
okay,
vision.
Yeah.
Oh, it would be cool, really.
So,
but you need the special glasses having such type of gene in your retina, right?
Yeah.
Concerning the patient experience, what would be
Yeah.
the patient experience? What do you do? Injections? I mean, how does it work?
Yeah, good question. Thank you. So the way it's injected is actually what's called an intravitreal injection, so into the center of the eye. It perhaps sounds a little scary on the surface, but it's actually done quite frequently, and there are products on the market that go by this route of administration now, and many retina specialists can do dozens of these injections in a single day. So the patient experience is really, it's going to be an outpatient experience. I would just add that the way that we go from the needle to the target, the cells that we target, is by a vector called a viral vector adeno-associated virus, and that's what actually carries the gene. And in the way that viruses behave, they transfer their DNA into cells, and that's what we take advantage of, if you will, in our gene therapy approach. So we inject into the center of the eye, the viral vectors transmit that DNA, and then over the course of days and weeks, that protein, this protein from algae that Dr. Pan mentioned, it's a light-sensitive protein that begins to express, and the expectation is that will confer new vision to people who are blind due to retinal diseases where the rods and cones die out in the eye.
How long does it take? I mean, after the product is injected, do you need just one procedure or few procedures? And how long does it take before the person would feel the light and would possibly see?
Yeah. Also great questions. Thank you. I think we're planning on a single procedure. And that's, yes, thank you. That's one of the advantages of gene therapies, really, is the expectation is that a single procedure can be the cure in many of these conditions. We know that it takes a few weeks for the protein to reach a peak expression level and plateau. And so that's out to about 8 weeks. And so the hope, the expectation is that by that time, our patients will be able to see some level of vision at that point.
What level of vision do you expect? How many percentages of the vision? What is your expectation?
Well, we're expecting to it's called transducing when the virus passes the DNA into those cells. So we're expecting to transduce about 10% of the what are called ganglion cells in the retina. These are normally involved in transmitting a signal to the brain, but due to Dr. Pen's wonderful work, we've been able to demonstrate that they can be given a second ability, which is to sense the light signal coming in. So with about 10% of the retina transduced, we think that we probably can't pick up the small numbers on the clock on the wall, but for somebody who's completely blind, things like seeing tables and chairs and other people, that's powerful and very meaningful. And so that's the expectation with our approach.
I would like the auditorium to put our hands together. I mean, that's amazing, in fact. I mean, that's unbelievable job. Basically, blind people can see after the procedure. Nevertheless, there are some limitations, Professor Pen, right? I mean, there are some things which it's still, for the moment, can't believe can be fixed with gene therapy, despite its gene defect. What are the limitations of gene therapy? When we already more or less understand that's a genetic problem there, but we cannot fix it. Would you please tell a couple of words about this one?
Okay, so now, also I should mention that, so for the gene therapy, basically they also can divide into two types. Okay, one is called a somatic gene therapy. So basically what it means somatic gene therapy means we're going to put the gene into the cell already differentiated, like in the organ, like eye. So that's the way right now we are doing this one, somatic gene. So basically we fix the people's blindness, but this one cannot transfer to the next generation. Okay, so because if the parent have a defect of a gene, the kids they're going to still have this one. So that's one. Now another group of gene therapy is called germline gene therapy. Basically, that's one going to correct or repair the gene in the DNA, in the sperm and the eggs. Okay, so before the fertilizer. Okay, so in that case, okay, then if we fix this one, then this is going to fix even the offspring. Okay, so that's the. So this is true. So for the somatic gene therapy, right now we have limitation is this one. You can only fix the individual and not their next generation. For the germline, we can do this one, but of course the germline we have more controversial, so we can talk about more this one.
Okay, I already make a part of provocative question concerning infrared vision, but if you tell about the complete somatic gene therapy overall, not only eye, but all the other problems and other issues. And that's, again, provocative one. Is it possible with somatic gene therapy, and where you see the perspective of this, to make human being better? To make it more strong, more healthy, less addictive to some stuff? Whereas somatic therapy has more perspective to move faster and to give us more advantages in our daily life?
I think, look at this one. So you means you want to some kind of enhancement rather than therapy. So
that's what I want.
Okay.
It's all of us want.
That's more fascinating. Okay. I think for this part, I think of course you can do the somatic gene therapy to do this one for the eye. So we can make this RG gene. RG gene basically is the gene right now we have is sent to the blue light. So wavelength of about 460. But people continue to try to modify to molecular engineer. We can make this protein going to send to the infrared light. Okay, shift this one. So if you inject this one, of course, the person can see in the dark. Okay, that's all. But I think the best way to do this one will be in the germline gene therapy. So that's one you can permanently make this person to be whatever you want. Okay, some people want to become more higher or want to more beautiful. Okay, the cosmetic industry probably will be very like this one, and also you can enhance people's muscle, become a very strong person. Yeah, I think this probably will be the gene therapy will be the best way to do.
And what is your expectation when this technology would be ready for the implementation? How many years it would take before we can really do this, approximately?
I think the technology already there. Of course, they have a risk at this point because when you insert the gene, whatever in the somatic or in the germline, you have risk to put in the wrong location that create a problem. Okay, but in general, this technology is there. But the only thing is, if that's really ethical issue, legal issue, or moral issue. Okay, this is basically we have to answer this one before we really can. Because right now, I think that the government do not even want to approve this one. So,
Okay, we will come to the regulation issues a little bit better.
people already made, scientists already made this one in the animal, in the mice. Okay, and they also made this, but
So
it's not in the
the same as in Tom and Jerry cartoons, there is a mice who is strong like an athlete. There is a mice. It's already done in the labs, right?
Yeah, they can do this one. Yeah,
Okay,
in the mice.
that's interesting. But still, I mean, on what you are saying, in fact, that technologically, we are ready to make this superpower man already now, with certain risks, but technologically it's possible.
I cannot say because in certain aspect, because still people study how to determine which gene we have to modify to make people become more stronger, or which gene so that. But if you know this one, that the technique already there. Yeah, so you can put in. Of course, they still have a risk. We need to further improve the technology.
Okay, it's getting more interesting to live, I would say. I mean, so we can wait next like 15 years, and it can change everything. That's not only about the physical feature, but it's again about longevity. That's about everything, in fact.
Yeah, that's one. But I think from my perspective, I more like I want to do this as a therapy instead of as an enhancement. That's probably something more controversial to think about. Yeah, and also I think this could be also change the practice of the medicine. So right now the medicine basically is mainly based on the treatment-based the medicine. But in the future, you could be this transfer to the prevention-based gene therapy. So if you already know the gene, you can fix in the beginning. So totally fix this one.
Okay, that's interesting. I mean,
if I can add a couple of comments, I would just say that certainly the techniques are available now and being worked in the lab and so forth. Translating that into humans is a very different story, and as I'm sure a lot of people in the audience can appreciate, that's it takes many years of hard work, and as Dr. Pan very rightly pointed out, I don't think that the government agencies are ready to contemplate that, and so don't want to bring the mood down, but I think that conceptually it's very interesting and compelling, but I think truly applications where you're looking to cure a disease is where things are at the moment, and
yes,
I think rightfully so.
I absolutely agree. But you see what Professor Pan mentioned, that's possibility. If you have the prediction that this certain gene can be fixed before the disease appears, that's already a medical question. That's enhancement, but at the same time it's a medical question. That's Angelina Jolie's story, right?
Yeah.
That's exactly Angelina Jolie's story. Everybody knows that she makes the operation because she made the genome sequencing. She found there is a certain defect on the gene which can come to the cancer later on, and she made the surgery. In this case, it's not needed, right? It can be fixed on the genome level. Wow, that's cool. But Professor Pan mentioned that there is some certain ethic questions. And Sean, may I ask you to comment on this one? We are talking about the government, but it's not only about the government. In fact, all the people sitting around, either scared, or maybe from the religious point of view, they are not ready to accept this new type of therapy because they are thinking that it's too rough interference in basic human nature. What do you think? If there is an ethic question, and how we can overcome this?
Yeah. Well, I think, from where I sit, it's a one step at a time type of approach. So we're working at present to get the first gene therapy approved. And by we, I mean the scientific community is working at present to get the first gene therapy approved in the United States. And so with, with some hope that'll happen next year. As far as taking it from that standpoint and moving into somatic cell, or germ cell type of approaches, I think we're still quite a ways off, and you're absolutely right. There's a lot of debate that needs to be still fleshed out as to whether it's ethically the right thing to do, and if you can prevent a child from having a disease before that child is born. That's a very big question, and I think it's something that we as a community, as a society, need to keep talking through.
Okay, but concerning the adult people, not about being a child.
Sure.
I'm coming back to enhancement, sorry. That's provocative, yes, but still, I mean, that's very interesting. You are on the edge of the technology. You know better than us how it works. You see that this technology is already available. But what about the ethic issues? I know that you are talking with a lot of people about this exactly point. What people in different societies, in different countries, what do they think about this one, and what is your personal opinion? Do we need to do this, the enhancement of the human nature?
My personal standpoint is in most cases, probably not. I think that diversity is a beautiful thing, and I think we should, remain a diverse population. It is an interesting question, though, when you start to divide the line between therapeutic and cosmetic. So, for example, building strength and muscle mass. There are some indications, diseases where that would be tremendously beneficial. But for most of us, it's probably not imperative. When you start thinking about athletes and professional sports, already we have laws against drugs, and in many ways, gene therapies are absolutely drugs. So, I think that gets into a kind of a slippery slope if we contemplate something like that, which is, largely cosmetic in nature. But I would just reiterate, we're focused on fixing, curing blindness, or at least restoring vision to the blind for the time being.
Okay, I do not agree that Association of Cosmetology Surgery would agree with you. Okay. They really believe they need to fix human nature and to make it better. Nevertheless, that's a new technology paradigm, and with these changes, I believe that it has to be some changes done in the approach of the state and the regulators to the technology. That means that the clinical trials for certain type of genome editing or genome somatic therapy has to be different. And how fast regulator moves this pathway, and what are the changes, what are the risks, and as well, like a simple one, how long has to be clinical trials if you are talking about genome therapy, because you need to see maybe second generation even.
So there has been a pretty major change in the approach to gene therapy from regulators over the last few years. And as Dr. Pan pointed out, in some of the earlier clinical studies, there were issues with safety with gene therapeutics that really forced the regulatory agencies to take a closer look at how these products are being developed. The major advances over the last decade or so have been in the way that these are delivered. So I mentioned that adeno-associated virus vector. And that has enabled a lot of these safety issues to be overcome. And now we've seen in the EU just a few years ago in 2012, the first gene therapeutic approach was approved called Glybera. It's by Unicure. And so with that, I think there's an increasing comfort level in the regulatory agencies, both in the EU, the US, and across the world. But indeed, we are at the precipice of seeing the very first gene therapeutic approved in the US, and at the precipice of seeing the first optogenetic therapy get into human clinical trials in the very near future.
Okay, so what you are saying, the clinical trial length more or less similar to the regular clinical trials where we have it for the small molecules or for biologicals, it would be more or less the same.
It is roughly the same.
Okay.
Still takes a good, probably 4 or 5 years to get a product through development.
And the cost for the clinical trials, would it be the same or would it be less, more? Because, I mean, again, we are talking about the economical multiplicators, and that's expensive to have any drug.
Yeah. Well, it partially will depend on the indication you're pursuing.
Sure.
With gene therapeutics, most of the indications are orphan diseases, so smaller patient populations and consequently typically smaller patient numbers. On a per patient basis, I think actually they're more expensive. And so if you start going into mass markets with a gene therapeutic, it's probably going to be a much more expensive clinical trial. But in orphan indications, it's given the smaller patient populations, it typically will translate to a lower hurdle in that regard.
Okay. You are the investor in this project. That means, being a businessman, you do expect return on investment in next 5 years, 7 years. How much does it take? Nevertheless, I suppose that the system of compensation for this type of treatment has to be a little bit different with the one you have now, and has to be some economical basis for the economical system of the country you are selling the new method of therapy to accept this drug and to pay for this drug.
Right.
How it's fixed and how it's working in genome therapy, because first of all, we are talking about the population sometimes didn't suppose any type of treatment, like blind people. It was just a cost for support the normal life for them.
Yeah.
And it's completely different payments, completely different budget, completely different approaches. Now you are moving them being a patient, which can be fixed, which can be treated. How the fiscal and financial system would approach this issue and would adopt this issue?
Sure. Great question. And I guess a few questions embedded in there. So I'll kind of take it from a high level and move down. The first thing I would say is that drug development is a very risky business, as I'm sure everyone here can appreciate. And the attrition rate in clinical trials is high. And moreover, the studies are expensive. So absolutely, there is a need to be able to recuperate the costs of the failures when you do have a success. And that does drive some of the pricing in drugs that we see. The interesting conundrum, if you will, in gene therapy is that it gets compared to other therapeutics where it might require a monthly injection. So I mentioned Lucentis, for example, that is a monthly injection, and it's $2,000 per injection, and it can readily, over the course of a few years, get very expensive. In the case of a gene therapeutic, it's one injection, but that therapeutic continues to be produced on a day-to-day basis. And so how do we compensate for that appropriately and build that in? And there's been a lot of discussion around that. The one therapeutic that is on the market is Glybera. That's in the EU, and it's currently being reimbursed at over $1 million for a single injection. If we compare that to other drugs that are, several hundred thousand a year, it actually works out not to be as expensive as it sounds on the sticker price. There is and has been some discussion about whether it makes sense for insurance companies to reimburse on a year-over-year basis for a gene therapeutic as opposed to one lump sum up front. Mm-hmm. And those are things that are yet to be worked out. We're just entering our phase 1 clinical study, so we're not there yet as a company, but that's definitely a space that we're paying close attention to.
But you mentioned a very interesting point. The gene therapy is the switcher. One injection, switch off, no disease, person is healthy, more or less. I'm a bit overdoing this, but anyway. What is the sense for the industry to go further with this type? Because with this type of the therapy, because the population with this type of disease is limited, more or less. What is interested for the industry come there?
Yeah, well, so in the U.S. there is what's called the Orphan Drug Act, and orphan diseases are characterized by patient populations of 200,000 or less. Mm-hmm. And so the Orphan Drug Act is designed to give incentives to pharmaceutical and biotech companies to develop treatments for those patient populations, because while individually they're small, if we add them all up, they're quite large. And if we took everyone in the world with an orphan indication and we put them into a single country, it would be the third largest country in the world. So collectively, these are issues that need to be addressed. And so the incentives in the US include orphan drug exclusivity, meaning that you can, once you're on the market, you can exclude others from coming in with that same product, basically, whether you've got patent protection or not. They include some incentives through the course of drug development, but then also incentives in tax breaks and so forth as you get through and approved. And so, it so far seems to be effective. There have been a number of companies that are now pursuing orphan indications. And
should the patent protection be longer for gene therapy then? Because due to the smaller population and due to complete treatment, you need longer period to cover your cost for the development. What do you think?
I would vote for that.
Okay, before I give the mic to the auditorium, I would like to ask you, Professor Pan, what is the next? Okay, next few days is very important for you. We really wish you the success with the first patient. We want to see this in newspaper as the first really successful case with the blindness treatment. But what would be the next for you? Which target next? Where you want to go with the gene therapy?
I think for my lifetime, I just want to continue improve the technology.
Okay.
Because actually, right now we are trying to target in the retina, in particular cell type, the retinal ganglion cell. Okay. So I think we try to target in another cell type. So try to continue to improve the resolution. That's the.
So from 10% to.
Yeah, I think the percentage of the cell, I think that can be further improved. And also we can do target in another cell type. So I think probably people, maybe some people don't know. So in the retina, they have a 3 layers of the neuron. Okay, the first layer is the photoreceptor. So that's the cell sending to light. Yep. Then they have a second order neuron, is called the bipolar cell. And then the third order neuron is called the ganglion cell. The ganglion cell is the final neuron, send the signal to the brain. Okay, so when the photoreceptor died due to this degenerative disease, okay, so then the rest of the cell, bipolar cell and the ganglion cell still survive. We have about 100 million photoreceptors. Then we have 10 million bipolar cell and about 1 million ganglion cell. So currently, the RetroSense right now, the first step is we put this gene into the ganglion cell. And so next step right now, we try to put it in the bipolar cell because the bipolar cell have 10 times more than the ganglion cell.
Is it different gene defect or the same gene defect?
The same gene. Just have to figure out use the virus can specifically target in the bipolar cell. Yeah, that's what we try to do.
Oh, that's cool. Okay, sorry, I switched to Russian for a moment. Коллеги, пожалуйста, вопросы из зала, если можно, микрофон на середину вынести, чтобы помочь. Вот, пожалуйста, вопрос. Идет.
Thank you very much for the very interesting talk. I have 2 questions. First one, I'm not sure whether you mentioned it or not, but do you vector insert into the DNA of the patient or not? So if yes, how do you prove that this is secure, that it will insert in not in a place where it could get cancer or some other not very good things? If not, what do you do with the loss of this vector in the term of cell divisions? And the second one, you mentioned the price of each injection. It's enormous, like several million dollars as far as I understand. Okay, why so expensive? Because to make vector, if you know what is it, it's can be easily done in more or less any lab in the world. So, thanks.
Sure. Do you want to take the first part? I'll take the second? Or?
Okay.
Okay.
Yeah, good divide. Okay, now the virus right now we are using is the adeno-associated virus. It's called AAV. So we package the gene into the. This is basically is a harmless virus. So basically, usually the virus, the white type of virus, when you put it into the cell, so basically they're going to duplicate. Then they're going to create more virus. They're going to eventually cause a problem. But in this recombinant, we basically get rid of the gene, which can prevent their continued duplication. But they're going to package our therapeutic gene. So this is like we are using from the green algae. So now the gene insert into for the AAV, as we know, this is they have a certain chance to insert into the genome, okay? But the chance is very low. So right now, basically, the gene when they go in, they like exist as like a small island out of the nucleus is called episomal, okay? Is there. So basically, this, as I know, this, so far we do not think this is a concern. Going to insert into the particular population could cause some problem, could be cause cancer or something.
And the other part of the question on the episome is whether it will divide out. And because we're treating end-stage differentiated cells, the ganglion cells, they don't undergo division, and therefore it's expected that the gene will remain and the expression will be continuous for many years, hopefully the life of the patient. And so to the second point, I would just make a correction. I mentioned Glybera that is $1 million, north of $1 million for a single injection, and that's in the EU. Glybera is for a condition called lipoprotein lipase deficiency. I wasn't suggesting, actually, I guess, that our gene therapy would be priced in that range. We still don't know where it'll be priced, but it's one model and one example of how the issue of perpetual therapeutic expression is priced. And beyond that, I would go back to the point that in drug development, it's a very high attrition rate. And there are very high, the costs of failure are very high, and those need to be recuperated in the ultimate price of the drugs that do succeed. So it's really, I think, looking at the cost to make a research grade batch of product in an academic lab at a bench is not really an appropriate baseline for what these should be priced at. And in the case of blindness, pharmacoeconomic studies show that the lifetime cost of someone with retinitis pigmentosa is about $500,000. And so, that's another way to look at it, is what value are we generating, not what does it cost us to bring a product, to manufacture a product at the bench. So there's a lot that goes into the pricing mix, and these are all things that do need to be considered.
Пожалуйста, еще вопросы, коллеги. Можно задавать вопросы по-русски, у нас есть перевод, если кто-то хочет задать. Окей. Интересно. That's interesting that the question was about the price. In fact, you see, I am not asking.
All right.
Dear colleagues, thank you very much for coming here. I really wish you success with your first patients. It would be breakthrough, definitely would be breakthrough with human therapy of blindness, with genome somatic therapy. And put your hands together. Давайте поаплодируем нашим гостям.
Thank you.
Thank you.
Thank you.
Thank you very much. Итак, у нас сегодня в гостях были профессор Джуон Хуа-Пан, специалист по генной терапии из Университета Уэйна, который, собственно говоря, смог заставить слепых прозреть, и господин Шон Эйнсворт, основатель и генеральный директор компании RetroSense Therapeutics, который поверил в идею, вложил свое время и свои деньги, чтобы это стало реальностью. Спасибо большое. Thank you very much.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you very much.
Thank you.